Okay, hi everybody. Today we are going to present the webinar of the dealing with the topic of the monkeypox resurgence. On August 14th, 2024, WHO declared Mpox a public health emergency of international concern due to a sharp rise in cases in the Democratic Republic of the Congo and neighboring countries, including Burundi, Kenya, Rwanda, and Uganda. This webinar aims to provide a comprehensive exploration of the recent resurgence of Mpox. Our objectives are to shortly explain clinical features, diagnosis, and treatment approaches, to explore the virology and review current vaccine options, to explain epidemiological trends and insights, and to summarize strategies to build resilience against future monkeypox outbreak. In fact, all our speakers today are eminent figures in their respective specialties. If I were to detail their qualifications and experience, each would deserve a separate webinar. The first speaker is Prof. Dr. Rodrigue Moral Alfonso. He will talk about clinical features, diagnosis, and the treatment approaches of Mpox. Please, Dr. Alfonso.
Okay, let's start. Thank you very much for the invitation, and I will start speaking about the clinical features, diagnosis, and treatment approaches. I don't have any conflict of interest, and this will be the outline of my presentation, a brief introduction being the first, clinical features, diagnosis, treatment, and some conclusions. As has been mentioned, Mpox is a disease, an old disease, in fact, emerging according to what we define it geographically, that is caused by the Mpox virus. It's a Pox virus in the family Poxviridae. This will be discussed later in detail of the genus or orthopox virus, where you have other important viruses, including especially smallpox. This is a DNA virus, as I mentioned. It was discovered in 1958 in Denmark, in monkeys, and the first human case years later in 1970 in the Democratic Republic of Congo. And important to mention that no human cases outside Africa occur until 2003. And this has been a neglected condition. There was a mistake here, not 23, but 2003 in the United States. This is the original report, just to remember that, in fact, this occurred in monkeys, but certainly, as I will mention regarding the stigma, it's important to remember to avoid the use of the term monkeys, as WHO mentioned, or instead of that, Mpox, and that he's been affecting especially African countries for a long time, since the 70s, especially children, especially in the Democratic Republic of Congo. And as we published last year in this systematic review, especially now in Africa and in the Democratic Republic of Congo, children are significantly affected by Mpox. But it's important, especially after the epidemics of 2022 outside Africa, to remember that not only men or children can be affected, but other populations, such as is the case of women and even elderly population, that can be affected by Mpox, as has been described. This has been a neglected condition, as I mentioned, in Africa, without the right support in terms of the development in research and development, for example, for antivirals and vaccines, as will be discussed later. And this takes importance, especially in 2003 with this famous outbreak in United States that has this clinical presentation that in fact affect children related to the illegal trade of animals, especially the prairie dogs that were in contact with other animals in Africa. And other cases later have been described, especially important. And after that, the research gained importance globally, as has been discussed in multiple analyses. At the International Journal of Infectious Disease, this was published at the start of the epidemic in 2022 regarding the underestimation of disease important. And in fact, after Mpox was declared a public health emergency of international concern in 2022, this gained really global visibility and importance in terms to address not only the topic of vaccines and antivirals, but particularly to understand the clinical presentation and new ways of transmission, as we may discuss later. In that way, it's important to mention that with this epidemics in 2022, this is the characteristic of a special an epidemic affecting mainly males in young sexual age, particularly in the group of the LGBTI community, especially men who have sex with men, especially also people living with HIV that we will discuss later, what happened when these populations are immunosuppressed. And this is part of multiple clinical characterizations that were made globally. In that way also, in this group of the LGBTI community, as I mentioned, the stigma occurred regarding this as occurred with other sexually transmitted infections. And in fact, regarding the name, as I mentioned, the World Health Organization changed the name for English to use Mpox instead of monkeypox. But even recently, we discussed this in Lancet a few days ago regarding the need to use correctly the translation of Mpox to other languages, for example, in Spanish, Viruela M, or in Portuguese, variola M, to avoid stigmatization regarding this disease that, in fact, obviously originated in animals, in monkeys, but is not only an animal disease. In some countries, for example, we experience it in Latin America. After the arrival in 2022, we start to organize clinical studies to understand the presentation of this disease in this. And this is part of one initiative of collaboration with colleagues in different parts of the world, IDID. And in this study, as you may see, regarding the demographics I already mentioned, it's important to mention that 100% of the patients present with rash with the lesion density tends to be, in fact, less than 50 lesions. The morphology regarding this distribution, a wide distribution of the localizations, and including other systemic symptoms, including in half of them fever, but as you may see, other presentations that we publish in this article in New Microbes and New Infection, including also lymphadenopathy, especially inguinal. And this is also important, as I will mention, during 2022 and beyond, the clinical presentation of this, especially was observed with genital ulcers. This has been characteristic in multiple countries, and also related to the close contact during sexual intercourse. As I mentioned, there are differences in immunosuppressed population, and this is important to remember that among immunosuppressed population, especially people living with HIV, there are differences in the clinical presentation and the risk, especially of complications and even fatal outcomes. This is a review we published in 2022, and here clearly described the classic presentation with the incubation period, later the invasion stage, the skin eruption, where initially during invasion, there occurred these clinical findings, including especially the fever, which is a prodromal one, malaise, asthenia, lymphadenopathy, and later in these infections, infectious skin eruption, the rash development, including all the possible skin lesions from the macule up to the pustule and cross, which is really important for diagnostic aspect, because from the skin lesions is where the virus presents the higher viral loads, and this is important for the PCR testing. There might be dissemination, primary and secondary viremia occurs, and this is also important for the multisystemic complications that may occur, especially immunosuppressed population. As you may see here, I will not enter in detail. We have to remember also about the pathophysiology and immunology, and this is part of recent review we published. And a typical presentation starts to be seen in 2022. For example, you may see this patient presenting initially with genital rash, even before fever and lymphadenopathy, and this was observed commonly regarding this. This become very important in the genital area for the 2022 and beyond outside Africa. I will not discuss on the clades that will be addressed later, but you will see these kind of lesions in different genital area. For example, lesions at the perianal region, also important. Even sometimes very few lesions that were may detect in certain areas, also important in this patient to suspect this possibility, especially in HIV patients, for example, with the anoscopic examination. And these lesions were reported in most of the series. Remembering that in those that were not immunosuppressed, the risk of complication were lower, as was reported, for example, in this series in New England Journal of Medicine, but with different timelines of presentation of these lesions. Remembering that lesions in the oral and perioral region may occur, obviously inside also a mucous area, perianal, anal, and rectal lesions. And as I mentioned in this publication, we made remember the oral lesions. Regarding the diagnosis, it's important to remember that there is a wide number of conditions where we have to keep in mind the differential diagnosis, including, for example, especially varicella, herpes, syphilis, among other vaccine-preventable diseases such as measles, or even the case of other sexually transmitted infections. In fact, as I mentioned, it's important to remember that Mpox can be detected in different secretions, including not only blood or for example the skin lesions, but semen, for example. But it's important to remember that regarding that skin lesions are the preferred to make the diagnosis. Remember that the diagnosis is made mainly by the PCR, but we can also perform experimental in some institutions other kinds of testing, including, for example, cell cultures and even serology that has been investigated. Remembering also that, as I mentioned, this has been detected in seminal fluid, for example, in semen. This is important considering that after that, this would be considered also in addition a sexual transmitted infection, but it's not the only way. It's important to remember that the main is the close contact, and in fact, the skin lesions are the main one for diagnostic tests. This was approved this week, a few days ago, and this is to remember that the WHO approves this first Mpox diagnostic test for emergency use. Is also a PCR-based test from out Alinity, and this is the first that has been approved. As I mentioned, to remember just that sexual transmission is also possible, co-infections with other STIs, such as the case of syphilis, and the patients as we publish in this case, may complicate it. May have, for example, secondary bacterial infections, but especially immunosuppressed population, even worse complication, in this case here in Colombia, of an encephalomyelitis in a patient with a lymphoproliferative disorder, but even cardiovascular alterations and severe skin lesions that has been observed in different cases, for example, in the United States and Brazil. And to remember that these cases even may evolve to a fatal outcome that has been especially important in HIV population, and this a study from Midja in Lancet demonstrated last year. In this context, it's also now concerning for the second time, the WHO declare again this as a public health emergency condition, and then it may bring new scenarios and challenge, including not only the risk of transmission, but what we need to understand from the clinical point of view, if there are really a difference between the clades and subclades regarding the clinical presentation. Regarding the treatment, this treatment in Mpox is mainly symptomatic according to the presentation or to the complications, and there have been since the 2022 the development of these clinical guidelines. But it's important to remember that we have been using antivirals such as the case of cidofovir or brincidofovir or tecovirimat, which are important, especially in immunosuppressed population for complicated cases that will be managed at hospital institutions. And there have been from these guidelines all the indications, the data regarding the efficacy and the safety that is available at the guideline of the WHO. This is the algorithm for management from WHO, which is also available in the guideline if you are interested to see, and to remember that those patients, especially under risk factors,
will be.
Sorry,
2 minutes are remaining. I'm sorry. Okay.
And the mic is still open. Sorry. Now, okay. And in addition to that, the best is to remember that it's important also to make prevention. I will not stop on this. You may later see this paper we published with Dr. Fatma Amer. And just to finish some conclusions from the challenge we have here, diagnostic difficulties as the PCR are not everywhere, treatment limitations that we have no currently common access in all the countries, for example, for these antivirals I mentioned. Later will be discussed the issue regarding the vaccines. I already mentioned the stigmatization and public health messaging is important right now to address topics such as the transmission dynamics and control to be better understand. And finally, to understand the impact on healthcare system that outbreaks of Mpox having, such as, for example, the case of right now, especially in African countries such as the DRC. Just to finish, I invite you to visit the special issue on monkeypox we have on our journal, Travel Medicine Infectious Disease, and thank you very much for your attention.
Now the second speaker is Prof. Manal Baddour. She will tell us about insights into virology and vaccine strategies for Mpox. Please go ahead, please, Prof. Manal.
I would first like to thank Professor Fatma for inviting me to share in this webinar, and I would like to thank all of you for attending. I will be talking about the Mpox virus and some vaccine strategies. So, okay. To begin with as Professor Alfonso said, the monkeypox was the original name because it was first isolated from monkeys in research labs in Denmark in 1958. The first human case was a 9-month-old boy in 1970. And Since 2023, the WHO recognized the change in the name or acknowledged a change in the name from monkeypox to Mpox because actually monkeys are not the primary reservoirs of the virus. And in fact, Mpox virus has a broader host tropism. And recent isolates from the current or the recent outbreak are different from genetically distinct from the original monkey-derived strains. And this name, the monkeypox name, implies that Africans are the source of the disease. Well, orthopox or Mpox virus belongs to the orthopox virus genus of Poxviridae. Within the orthopox genus, there are several other members, including vaccinia virus, which is responsible for, or which is used in vaccines, cowpox and others, and of course, the variola virus causing smallpox. Mpox is an ovoid or cuboid virus with 200 nanometer diameter and about 300 nanometer length. It is surrounded by inner and outer membranes, which serve as virus envelope, protein spikes, which serve as virus attachment proteins, and a characteristic internal dumbbell-shaped nucleocapsid. We will talk about it. And it also contains lateral bodies, which contain effector proteins that are released by the virus in the host cell upon infection to modulate the host cell function. Based on genomic analysis, the virus, the Mpox, can be differentiated into two main clades. Clade 1, which is also known as the Congo Basin clade, and clade 2, which is also known as the West African clade. Both of them are subdivided into 1A, 1B, and 2A, 2B, respectively. The genome of the virus is a linear, double-stranded DNA genome, about 197 to 211 kilobase pairs in length. And there is, it encodes about, it has about 193 open reading frames, which encode about 190 putative genes. The sequence similarity between clade 1 and clade 2 is about 95 to 96%. The DNA contains a central conserved region flanked by lateral variable regions of inverted terminal repeats and with closed hairpin ends. It's notable to, it's noteworthy to say that there is a terminal deletion of about 2 kilobase pairs on the right-hand side in the clade 2 virus. So some genes, like these 4 genes, are missing in clade 2, whereas these 3 genes are truncated in clade 2. If we look at the functions of these genes, we will see that many of them are concerned with immune evasion and host cell modulation. Together, these might be the reason why there is a difference in clinical presentation from clade 1, between clade 1 and clade 2. But having said that, there are also some genes found in clade 2 and absent from clade 1. The differences between clade 1 and clade 2, the mortality rate of persons infected with clade 1 might reach up to 10%, whereas in clade 2, it's less than 1%. The geographic distribution, as we said, clade 1 in Central Africa, whereas clade 2 in West Africa. Symptoms tend to be more severe with clade 1 infections, requiring hospitalization in many cases, whereas there are milder symptoms in clade 2 infections. Clade 1 is originally, or was originally zoonotically transmitted with spillover from animals to humans, with less capability of human-to-human transmission, but clade 2 has adopted or adapted to better human-to-human transmission. Vaccine is effective similarly in both clades because it utilizes conserved targets. And the viral load in infected individuals tends to be higher in clade 1, and this reflects on more severe clinical conditions versus clade 2. As said, the recent global outbreak in 2022 is primarily caused by clade 2B, which has appears to be adapting to more human-to-human transmission with less virulence. Other than disease severity, there are no clinical differences between the two clades. The incubation period starts about 5 to 21 days, and the major clinical presentation is the skin lesions found in about 95% of cases, with other manifestations such as fever. Lymphadenopathy is noted, and it seems to be diagnostic also, fatigue, myalgia, and headache. Rash of Mpox virus. The rash goes through stages, starting with macules, papules, vesicles, pseudopustules, and scabs. I will come to scabs in a few minutes. Not all cases go through all the stages, and the differences, there are a lot of differences in the numbers. If we look at the replication cycle of Mpox, it starts by infection with intracellular mature virus particles or extracellular envelope released. The intracellular mature virus particles are released from the host cell by cell lysis. The extracellular envelope viruses are released by exocytosis. Each of them, they bind to the receptors on the host cell. Internalization, uncoating, transcription, early transcription, intermediate transcription, late transcription with formation of proteins, then assembly of the virus particle, morphogenesis, formation of the intracellular mature virus particle, acquisition of an envelope from the Golgi apparatus, with formation of intracellular enveloped virus particle, which is released from the host cell by exocytosis and remains attached to the host cell, forming cell-associated enveloped virus, or is released forming an extracellular enveloped virus. During the replication, the virus encodes several proteins which act as regulators, whether internal regulators, intercellular or extracellular regulators, such as creating an antiviral state, blocking an antiviral state in the host cell, suppressing apoptosis, and regulating signals in the host cell. Extracellular regulators include inhibitors of cytokine and modifiers of host response. Just to give an example of immune evasion practiced by Mpox virus, the virus-infected cell presents the virus antigens on its surface in association with MHC1 protein, and these are recognized by T cell receptors on the surface of CD8 cytotoxic T cells. The virus inhibits the internal trafficking of MHC class 2, and there are many regulatory functions ongoing in between the infected host cell and the virus, but there is needed a co-recognition of a ligand found on the host infected cell, the natural killer G2D ligand, which is recognized by a natural killer G2D receptor on the host on the effector cell, the CD8 cell, and this co-recognition is needed for activation and the following cascade. The virus encodes a natural killer G2D decoy soluble protein that blocks the receptors so that they are not activated, and thus the following cascade is not activated. It also produces several proteins, soluble proteins, that block other effectors. For example, the C gene of the virus produces a protein that reduces activation of immune cells. Other genes produce proteins that target cytokines and chemokines and blocks them. Others block activation of the complement cascade. Others block the pattern recognition receptors signaling cascade. Other gene products prevent interferon alpha, beta, and beta signaling. Other proteins make the host cell escape apoptosis, thus producing more and more virus. Other genes prevent nuclear factor kappa B activation. Why are we seeing more and more outbreaks of Mpox virus? Because of increased human and to animal interactions, because of the waning immunity, because of cessation of smallpox vaccination, and due also to urbanization and deforestation. increasing the interaction between potential animal reservoirs which lost their habitats and encroached upon human residents and humans. Though the natural reservoirs of the virus remain elusive, however, there are believed that some rodents which share ecological niche overlap with the virus are primary reservoirs, such as rope squirrels, which are arboreal squirrels living on trees, Gambian pouched rats, dormice, red-legged sun squirrels, and non-human primates such as monkeys. Domestic carnivores, dogs and cats, serve also as secondary hosts, can be infected and can infect. Causes of death include bacterial infections of the severe skin lesions, pneumonia leading to respiratory failure, encephalitis, and severe inflammatory response syndrome. Transmission pathways will be mentioned. They are multifaceted, but I would like to point to the environmental contamination, which means that there is a big role for mites. The virus can infect, Mpox can infect a broad range of immune cells, such as macrophages, dendritic cells, and B lymphocytes. It can also infect epithelial cells and endothelial cells from the mucous membranes. And this serves to spread the virus, and it primarily targets the skin, the lymphoid tissues, and the respiratory tract. The host cell receptors of the virus are believed to be, the primary receptors are believed to be glycosaminoglycans, such as chondroitin sulfate and heparan sulfate, which bind the virus attachment proteins and lead to binding and entry of the virus. Laminins can also act as host cell receptors. Back to scabs. Scabs can be contagious even after they have crusted over. The CDC thus advises that infected individuals should remain isolated and not come in contact, especially with immune-compromised persons, until all the lesions have crusted and the scabs have fallen off and a new layer of skin has formed underneath. The virus is not known to infect intact skin, but it can infect intact mucous membranes. So how long can it survive on surfaces? On non-porous surfaces such as glass and stainless steel, it can remain viable for up to 5 days. On plastic surfaces, up to 3 days. On porous materials such as cardboard and wood, it can remain viable for 1 to 2 days. However, the presence of virus in dark, cool, and low humidity environments can lead to survival up to 15 days, and especially on linens and clothing. Thus, to eliminate the virus from mites, we need to use disinfectants, especially EPA registered disinfectants that are listed in list Q, such as alcohols and chlorine. But noteworthy that any disinfection has to be preceded by proper cleaning, because the scabs and organic material can shield the virus and make it difficult to eliminate. Thus, the best decontamination practices include use of appropriate disinfectants, preceded by regular cleaning, proper hand hygiene, and training and increasing awareness of this. In the lab, the virus can be diagnosed by electron microscopy or by immunohistochemical methods, detecting the virus antigens, or by serology detection, detecting the antibodies of the virus. However, all these are non-specific, and the virus should be, the infection should be confirmed by real-time PCR using different genes that have been used in real-time PCR, or next-generation sequencing, or even virus isolation, which is not available in many areas. I also need to stress on that before the onset of clinical symptoms, the virus or the PCR is unlikely to be positive. Now we shift gear to vaccines. A few notes. There are no currently developed specific Mpox vaccines. The vaccines in use are repurposed smallpox vaccines, and mass vaccination is not justified Only post-exposure prophylaxis for close contacts of a confirmed case are recommended, and a vaccine recipient would be considered fully vaccinated 2 weeks after receiving the second or the final dose of the vaccine. The vaccines currently available are Jynneos, ACAM 2000, and LC16. Jynneos, or the modified Vaccinia Ankara Bolivian Nordic strain of Vaccinia virus, is a live, non-replicating Vaccinia virus given in 2 doses, 28 days apart, given intradermally or subcutaneously. However, after 2 doses, the vaccine efficacy is said to be about 80%. However, some persons who have been fully vaccinated with the 2 doses did develop Mpox, though in a milder clinical presentation, so it's not fully safe or protective. ACAM 2000 is another live but replication competent Vaccinia virus vaccine. It is not currently of favor because of the serious side effects it causes, and we will mention them. LC16 is another replicating vaccine. ACAM 2000 is given by scarification. LC16 and Jynneos are given by intradermal or subcutaneous injections. And it's also LC16 is a replicating Vaccinia virus.
Prof. Manal, please, 2 minutes are remaining.
Okay. It is considered 3rd generation with fewer side effects. However, it's only licensed in Japan currently. Side effects of the vaccines include injection site reactions such as pain, redness, swelling, induration, itching, systemic reactions such as muscle aches, headache, fatigue, nausea, chills, and fever. However, serious side effects posed by ACAM 2000 include myocarditis and pericarditis, swelling of the brain and spinal cord, infections at the vaccination site, spread of the vaccine virus since it's replicating, accidental infection of the eye might lead to blindness, though very rare, severe allergic reactions in apoptotic individuals. mRNA vaccines are known for their rapidity, relatively fewer steps and lesser costs, and faster development of the effect. Vaccines, mRNA vaccines in development of for Mpox include mRNA 1769 by Moderna, BNT 166 by BioNTech, which is in partnership with the Coalition for Epidemic Preparedness Innovations. Both of them are in clinical trials and showing promise. Both of them utilize also lipid nanoparticles, which give advantages such as encapsulation and protection, efficient cellular uptake, enhanced stability, controlled release, and targeted delivery. DNA vaccines are also in development. However, they haven't reached clinical trials because of mainly funding issues. TNX 801 by Tonix Pharmaceuticals is a live attenuated horsepox Mpox vaccine made of horsepox attenuated plus recombinant DNA. And currently there are about 17 Mpox vaccine trials ongoing. 9 of them are on Jynneos, LC16-3, 1 trial for each of them, mRNA vaccines, and ACAM 2000. Thank you very much.
Thank you, Prof. Manal, for this. You have provided us with valuable knowledge on Mpox virology and vaccine strategies. Now I have to introduce the third speaker. Please, Dr. Jonathan, who is an exception to what I mentioned earlier regarding time constraints. Dr. Jonathan is an early career professional, and I feel compelled to share a bit about him. This is not only due to his impressive expertise, but also because he is based in Uganda. Uganda, a key region that witnessed the early phases of Mpox emergent, our resurgence, offering a unique and invaluable perspective. Dr. Jonathan is graduated from a career university, a very famous university in Uganda, known of having wide community services, and is using the One Health as an important tool for solving many problems. Please go ahead, Dr. Jonathan.
Thank you very much, Professor Fatma, for welcoming me. And I would like to welcome everyone who has joined us today. I'll delve into the epidemiological trends and insights of Mpox that we are currently that we are currently tackling. That will be my outline for today. I'll delve a lot on the global situation. I'll also delve on the focus on Africa because my colleagues have handled most of the things about how the virus started and how it has evolved over the years. So this virus, as most of the speakers have said, belongs to the same genus as the variola virus, which caused the smallpox epidemic or pandemic that we had several years ago. However, this Mpox virus has two distinct clades or subtypes, and each of these clades has subclades A and B, as I will show you later on how these clades are distributed worldwide and in Africa. Clade 1 having a higher case fatality rate compared to clade 2. The current outbreak that we have is basically due to the clade 1B, which is highly fatal, as I'll show you as we continue. So my colleagues have gone over this, but as of today, Mpox cases have continued to rise, especially on the African continent. And ever since the 2022 epidemic, we had over 120 countries have been affected up to date, with over 100,000 cases. The ongoing surges, as I've said, are basically, we had many of them in DRC, where we had an emergence of a newer strain, the clade 1B, and we've seen this new subclade spreading beyond Africa up to India, Thailand, and Sweden. To date, we've had several deaths, and Africa alone accounts for about 537 deaths, with significant transmission ongoing in Burundi, DRC, Kenya, Rwanda, and Uganda on the African continent. This slide summarizes the current global situation, looking at all the way from 2022 to 2024, though I'm going to dwell so much onto the 2024 data. As we can see that since August 2024, 48 countries are reporting cases of Mpox, and we've had over 106,310 confirmed cases, and of these, 234 have been reported confirmed under death. So looking at the different epidemiologic curves of the different regions, Africa, comparing it to other regions, we see that cases are continuing to rise in the African region. And this is the second in place is the Western Pacific region. You also see that cases are also rising, though not as steadily as the ones in Africa. So Africa alone is reporting the cases are decreasing in other regions. However, in Africa and Western Pacific regions, we see a surge in these cases. Looking at the global map, where are these Mpox cases? So as of August 2024, we saw a 15.6% increase in the new cases compared to previous months. And most of these cases came from Africa. Africa contributes about 62.3% of these cases. On August 15th, Sweden reported its first case. So looking at these maps, we try to see the confirmed cases where we see the deep, this is purple, or the deep blues, as someone may say. We see the DR Congo having over 10,000 cases, confirmed cases. Looking at the Mpox death, where are they most? Still, the DRC or Congo has quite a number of cases, as well as some countries in the Northern Americas. When we profile these cases, we want to also know, as epidemiologists, what are the key characteristics of these cases. We see that many of these cases, over 85% of them, are men who have sex with men. 52% are persons living with HIV, 4% are healthcare workers, and 84% is sexual transmission, meaning it accounts for the #1 leading form of transmission. So this curve tries to summarize the different transmission types. We see the sexual transmission leading, followed by human-to-human contact. However, to interest you, we also have healthcare-associated cases that are being reported, animal to human, which tries to emphasize the role of One Health in this current epidemic. When we look at the cases, we see that majority of our cases are males. And when we compare on the different regions, looking at the different cases and the distribution per gender or sex, we see that the demographic distribution of Mpox cases in African region are a bit balanced compared to other regions which are reporting a lot of many cases are male. So my other panelists have talked about the Mpox, how it presents, but as an epidemic is driven by a case definition, so that we know what is a suspected case and what is a confirmed case, because not every case will be Mpox. So the definition, according to the WHO, some countries have come up with definitions to suit their context. And in DRC, a confirmed case is any case for which clinical and epidemiological diagnosis of Mpox has been confirmed in a laboratory. So you may see that some of the cases that we are reporting, some are combined values of both suspected and confirmed cases. And these are some of the presentations that we are seeing in the African continent. We see the different distribution of the Mpox virus, whereby it is also affecting children because there is a mother-to-child transmission. So this is a 9-month-old child in DRC, and we see a case from Gabon, and this was one from DRC. These two are from DRC. When we look at the different clades, because as I said, the Mpox virus being a DNA virus, as one of you asked in the chat, has undergone mutations, and we see the different clades and where are these clades. The current clade that is responsible for the recent announcement by the WHO that we're now having a public health emergency was the clade 1B, as we can see cases in DRC speeding over to Burundi, some cases in India and Thailand, and a case in Sweden, that is in Europe. So I'm not going to delve into these because my other presenters have talked about how these clades are distributed and where they came from, but maybe to highlight that the clade 1A is spreading basically is basically linked to spillovers from animals to humans, meaning there are individuals who are consuming wild game, as I'll highlight some of the cases I profiled. And then the clade 1B is basically due to person-to-person transmission. And I've said the clade 1 is known to be more transmissible and causes more severe infections compared to the clade 2. Let us look at Africa in 2024. And as of 6th October, that is about 2 days ago or 3 days ago, we had Africa alone report having a number of confirmed cases about 6,754, and most of these were coming from DRC and Burundi. The number of deaths overall in Africa were 32, Democratic Republic of Congo contributing 25 cases. And we see that the most affected countries in Africa continue to be DRC with very many cases, followed by Burundi and Nigeria. The two maps are showing us how the confirmed cases of Mpox are being reported in the past 6 weeks as of 29th September. We see that many of the cases in red, 200 plus cases, are from the DRC. And then this other map highlights the other African countries that are currently having Mpox and at what levels, or how many cases we are seeing. The epidemiological curve for Africa, as you can see, we have the cases rising on the continent, and as of 29 September, 14 countries have reported Mpox cases and are having ongoing active outbreak. When we compare two countries, as I said, DRC and Burundi, we see that these two countries alone account for over 95% of the new cases, Burundi in light blue and DRC in red. So we see the cases in DRC as starting to fall, but the cases in Burundi are rising. So of the new cases, seven countries so far have reported, and Burundi, Liberia, Cote d'Ivoire, Central African Republic, Rwanda, and Uganda are where we find these cases. And the clade distribution of the recent countries the new countries that have reported, these were due to clade 1A, whereas India and Morocco reported clade 1B. So as I've said, we have challenges that are really limiting the diagnostic capacity, especially in the African continent. Thus, most of the cases, suspected cases, do not continue to be confirmed in a laboratory or may even die before being confirmed. So we have underreporting somehow. DRC, as a country that has been affected as of 29th, had 25 deaths. Overall, in the entire outbreak since 2022 has reported 990 deaths, and we see its epidemiological curve trying to, it's a sigmoid. So there's an increase in the cases over years from 2023. And among these suspected cases, the case fatality rate is concerning due to limited healthcare access. Most of the cases do not reach the hospital, as I'll show some challenges later on. And DRC has all the clades, both clade 2, and most of the cases currently are due to 1A and 1B. Cases being tested, about 40% of the suspected cases are being tested and report a 50% positivity rate. Another interesting finding is that majority of these cases are below 15 years, meaning children are highly impacted by this outbreak of Mpox. These account for alone 66% of the cases and 82% of all the deaths that have occurred in DRC. Majority of these cases are male, 73%, and we've seen two regions that have been impacted by this pandemic, that is North Kivu and South Kivu. In this presentation, I'll try to highlight some of the findings from North Kivu. So North Kivu is somewhere here. This is a DRC map. And as of 28th, 1,108 suspected cases were tested with a 29% positivity rate. 2 deaths were reported, case fatality ratio of 0.6%. And in that province of North Kivu, the affected regions are Goma, Karisimbi, and Nyiragongo. Goma has an IDP camp, that is where we have a refugee settlement. Therefore, we've seen several cases in the IDP camps. Among the confirmed cases, 36% were individuals staying in these IDP camps and have acted as hotspots, with several of the IDP camps reporting multiple cases. Sexual transmission continues to lead the mode of transmission and community spread as well. So in the IDP camp, 75% of the cases are children between 0 to 17 years, and they're evenly distributed between the males and females. These two curves try to show us that in North Kivu, the IDP settings, most of the cases are children, if you look at below here. So we see an even distribution between the males and the females. And these are cases in other places which do not have camps. As of October 5th, vaccination has started in North Kivu. So looking at age-specific incidence in DRC and looking at this curve, as I've said, children under 15 represent majority of the cases and death. Infants and children under 5 are at a higher risk of severe disease and death. Given the fact that these children may suffer from other skin illnesses such as chickenpox, measles, and rubella, we may have some underreporting due to delayed diagnosis. And IDP camps like these have conditions of overcrowding and limited resources for hand washing. Therefore, we see this may explain the human-to-human transmission through direct contact. Uganda, where I'm coming from, we see it is neighboring Congo. DRC is just a country here. Reported its first case on 24th of July. And this was in this district, which is a border district. It is a district that suffers from several things. Recently, they had a flood, influx of victims of the civil war going on in Congo by the ADF rebels, causes people to come to Uganda, which hosts a very large refugee community in the entire world. So Uganda, as of 3rd October, did not report any death. 20 cases were admitted and reported 24 recoveries. So no new cases were reported in the last 24 hours as of 3rd of October, and we see that majority of the cases are Ugandans, with 3.6% being non-Ugandans. We can see the distribution. We see more males being affected in Uganda. And these are the districts. 13 districts have confirmed Mpox cases. We may see that most of the patients or the cases have history of travel from DRC Congo into Uganda, or history of staying in this district, which is a border district. That is how these cases ended up in Kampala, the capital city of Uganda, and some in the northern part of the country. I'll choose one case from this. This is Uganda. The first case was a 37-year-old female from Congo who was pregnant at 21 weeks. Stayed in Kasese, had a non-pruritic generalized papular skin rash eruption that involved her hands, and she reported resolution of these symptoms within 1 week. And what we see, she's a frequent traveler to the DRC, where the current epidemic or outbreak started, and has history of consuming various meats, including wild game. So Uganda's efforts currently are focused on tracking down the known contacts and improving surveillance, among other things. What are some of the challenges that we are seeing in responding to Mpox? There is limited testing and surveillance, whereby we lack point of care testing and laboratory capacity in many of the affected areas. So we have to see that wherever there's a case that is being identified, samples have to be collected and transported. The laboratory in Uganda, as for example, that is doing the sequencing is the UVRI, Uganda Viral Research Institute, relies on samples being transported to it from the different parts of the country. And then as the pandemic goes on, we have a vaccine supply constraints. The previous speaker mentioned about that the current vaccine vaccines are actually replicas of vaccines that we used during the smallpox and other outbreaks. So most of the countries that are affected are low-income countries and heavily rely on donations from high-income countries. Therefore, there is we may see a lot of inequity in terms of distribution of resources during this pandemic. Another thing that comes up is stigma and overlapping risks, because as I showed you in the profile of the cases, that the HIV infected population or people who are immunocompromised are also leading in the cases that we are seeing on the African continent. So now having Mpox giving the different skin lesions and HIV, that is a double burden. So people with Mpox and HIV are likely to face more stigma, and this may hinder timely access to hospitals and treatment. So these cases may stay in community, and then we see more cases going on. There is currently limited access to healthcare in many parts of the countries that are affected, and the health workforce is restrained or is constrained with resources and fear of being vulnerable. As we have seen, some of the new cases are healthcare workers who might have contracted this disease while taking care of Mpox patients. There are also competing priorities. Countries that are having the pandemic currently face multiple humanitarian crises. As I say, the DRC is having several rebel groups such as the M23 and ADF, which continue to block access to communities that are affected by Mpox through blocking roads, attacking communities, and this hinders really even WHO and other partners who may want to deliver supplies to these areas. Find it a big challenge to do that. Travel restrictions are not so much or have not yet been implemented. Therefore, the outbreak may continue to spread on the continent and out of the continent. This may explain the recent spillage of cases to countries outside the Africa region, like Sweden, Thailand, and India. The case in Sweden had history of travel to DRC, which shows that countries need to heighten or to increase on their surveillance and readiness to receive any new returnees from countries that are affected by Mpox. As I conclude, we eradicated smallpox several years ago, and the eradication demonstrated the power of coordinated international action. Therefore, I think for us to be able to eliminate Mpox, we must replicate the lessons and successes that we got through a united global effort. Some of the strategies that can help us achieve this include enhanced surveillance and data sharing, equitable access to medical countermeasures, including equitable access to vaccines. We shouldn't wait for the pandemic to go on and wait for the high-income countries giving the low-income countries leftover vaccines, as we saw during the COVID-19 pandemic. Several countries registered expiry of vaccines, and some of them were just dumping them in these other countries. So we need a coordinated effort to that. We need strengthened healthcare systems that include the One Health approach, since we have seen the interaction between the animal, environment, and human interfaces. We've seen that the cases have history of consuming wild game, which are right now, as we saw, the monkeys being from time immemorial, 1958, when we had the first discovery of this virus. Community engagement and education continue to be key. We need to educate the communities on how to identify lesions and on how to handle people who are sick. In Uganda, I've observed most of the cases that are suspected are not quarantined because of lack of resources. So they are let back into communities and asked to promptly report on how they are feeling. That is not really reliable. So we need to educate the community and the people about this. We need to invest in research and innovation so that we can develop newer medications and vaccines. Finally, we need to work together so that we can overcome challenges that Mpox has brought our way as we build a healthier future for all. I would like to thank everyone for listening to me. Over to you, Professor Fatma.
Thank you, Dr. Janassen, for sharing your valuable insights and expertise. Your perspective, especially from the context of Uganda, has been truly enlightening. We greatly appreciate your contribution. Now the fourth speaker, after we knew many aspects, important aspects related to Mpox, it's crucial to explore how we can combat it, along with the role of resilience and the One Health approach in this effort. Our fourth speaker is Professor Heba Mohtady. We'll delve into these topics, providing insights into effective strategies and the importance of a holistic integrated response. Please, Dr. Heba.
Thank you, Professor Fatma, for inviting me for this insightful and interesting webinar. I'm so excited to be with you here, and I'm interested listening to the presenters in this webinar. I'm talking now about Mpox from different perspective, about enhancing resilience through one approach or one health approach. So before I'm going to start, I would like to say that I am deeply passionate about this topic because it reflects the connection between my previous experiences and qualifications in different perspectives. I would like to share a secret with you is that while many people have hobbies like gardening or collecting the stamps, I have a quite different hobby. Thus, I prefer navigating and exploring different disciplines from medicine to education to management and leadership. And that's why I would like to talk today about the topic that is covering, One Health is covering my experience in medical microbiology and interest in antimicrobial resistance, and my work and PhD in healthcare disaster resilience, and my medical education leadership. So that's why I'm so interested about talking about this topic. What would be the aim of the presentation? How can resilience, particularly for healthcare systems, be enhanced through the One Health approach? I'm happy that Dr. Jonathan mentioned that there is a problem in healthcare systems regarding Mpox, and this is why we are going to talk about this today. Before we dive deeply and profoundly in the topic, this is our roadmap today, and it wouldn't be so long. So let us start by introducing One Health approach. The concept of One Health approach is that it integrates the health of people, animals, and ecosystems into a unified approach. It emphasizes that the health of humans and the environment are closely linked. So the main or the key aspects of One Health approach is that collaboration across sectors, focusing on health challenges, global application, and sustainable development. Human health and its interconnection with others like animal health and environment health. If you can see this picture, and you can see that the climate change, this is an example of the environment health, how can be affected and how can it affects human health in different ways. For example, climate-related hazards, extreme weather, heat, sea level rise, or whatever, they can all result in different health outcomes, including what we are talking today about, which is zoonosis and vector-borne diseases and malnutrition and several, even mental health is highly affected in several people by the climate change problems. The health systems are affected by climate change. That's why we would like to talk about each of these later on. Now, what about the animal health, the zoonotic diseases? It lies at the intersection of climate change and human health. So many of the 21st century's critical health threats are zoonotic. And environmental changes, such as climate change and deforestation, can alter the habitats of disease vectors and hosts, leading to new patterns of zoonotic disease transmission. And Mpox that we are talking about today is an example for changes in zoonosis and in different One Health approach. So now let us go to the resilience. What does it mean? And please look at this photo because it means a lot. If you can see this boy throwing the ball, it was used to talk about resilience as bounce back, exactly as this ball is thrown by the boy and bounce back to the same situation. It means like if the system is exposed to crisis and then it became, after that, in the same situation before this crisis, it bounced back. However, if this boy has an egg in his other hand and throws this egg on the ground, it wouldn't bounce back like the ball. And that's why the ball is resilient, our metaphor for resilience, and the egg is a metaphor for being not resilient. The expression I mentioned is just a metaphor. However, the definition is the ability of a system, community, or society exposed to hazards to resist, absorb, accommodate, adapt to, or transform and recover from the effects of a hazard in a timely and efficient manner. And this part is very important, including preservation and restoration of its essential basic structure and functions through risk management. This photo is someone who is falling forward. And now some people said like resilience is not just bouncing back. It's the art of falling forward. And this is a kind of evolving in the definition. And instead of just return to our normal situation after crisis, it means that people who are resilient, they can transform and grow and become more strong or stronger after such situations. So what about during infectious diseases? What could happen? Infectious diseases, outbreaks, pandemics, and other natural and human disasters, they all strain healthcare systems. However, people tend to expect that hospitals should be always available, functional, and accessible to meet the increased demands. However, in the reality, hospitals sometimes can't do that, and we have well-documented cases of hospitals that couldn't be capable of performing properly during or after disasters. This reflects the significance of healthcare resilience. And if you can see in this image, you'll find that all the surrounding is talking about the policy, the health, the surveillance, and the healthcare systems and integrated digitalization. All of that is mentioned about the healthcare resilience. Also, WHO mentioned that while resilience is important, and here developing resilient health systems ensures countries can effectively prevent, prepare for, or detect, or adapt to, respond to, and recover from public health stress while ensuring the maintenance of quality, essential, and routine health services in all contexts. So over the last 4 years, I have extensively explored this topic in my research and had several published journal practical contributions that included decision support tools, organizational learning tools, staff empowerment tools, and overarching disaster resilience tools. Also, I contributed to the expert meeting in the EMRO by the WHO, and this result after 3 years of work into the Resilient Hospital Guide, which is an interregional guidance. Let us go to application of this knowledge on Mpox response plan. Mpox is increasing even in Australia, and there is a rise about 570% since July, but the vaccination rates remain low. And as we know, and you kindly mentioned that it's in Africa now, there is the Africa CDC and WHO launched a joint continental response plan to strengthen and accelerate efforts across Africa in combating the ongoing Mpox outbreak. This initiative focuses on controlling the virus spread, saving lives, and protecting communities. These are the components of the plan. And these components are very important when we think about this plan because they should be supporting the situation and the society and the healthcare system to be resilient. Let us connect the dots between these items, I will write them again, and the resilience. Here in the plan, the Mpox Continental Preparedness and Response Plan, in the first page, it's written that fragile healthcare systems are inadequate. Funding and inadequate funding exacerbate these issues. We published a paper about investigating organizational learning and adaptation for improved disaster response towards resilient hospitals, an integrative literature review. And in this paper, we developed a hybrid resilience learning framework for evaluating resilience and organizational learning following disasters. I'm going to compare the components of this HRLF and the plan just I mentioned about Mpox. So these are the items of the plan or the components of the plan, and this is the areas of learning developed in this resilience framework. And you can see the similarities in numbers. I added the numbers from the components of the plan to this figure. And this indicates that the work done now in this preparedness and response plan is aiming to support the healthcare systems and the community with what is required for resilience. Again, talking about leaders and staff empowerment tools. Staff in healthcare systems are very important, are the assets of healthcare systems. And you can see like the resilience and adaptation for human health and animal health and environment health. The resilience and adaptation should be applied here on health workforce because they are human beings and they are exposed to a lot of factors of stress and lack of resources and many other issues. That's why we developed in another published paper, Healthcare Workers Resilience Toolkit. In these skills, we focused on their needs like mental and physical needs, education, resources, and communication. Let me go back to our situation, which is the One Health approach. One Health approach is something that require communication, collaboration, and coordination. And that's exactly as we mentioned in the resilience framework and in the resilient toolkit. Again, in this proposed healthcare worker resilient kit, education was a cornerstone, and it has been recommended to empower our health professionals and healthcare workers with different ways of training, courses, programs in different ways. But if you can see, it's multi-professionalism, like interprofessional collaboration. And also it increases the focus of these healthcare workers on the climate impact related problems. Why I'm saying that? I'm saying that because we need that our staff be informed with the component of One Health, and it includes these issues. Also, the leadership is very important, enabling transformational leadership to foster disaster-resilient hospitals. In this publication, a model called APRA model was developed. And in this model, resilience was included in several actions, like anticipate and assess risk, of course, and then prevent to prepare, respond, recover, reflect and regenerate, and finally apply all what have been learned from this cycle and assess again. And this reassessment is important for continuation of the process. And these are the capacities required for resilience: the anticipative capacity, the preventive one, the absorptive one, the adaptive one, and finally the transformative one. Now, thinking about transformation in One Health approach, again, this is the transformation required by One Health approach. You can see how the dots are connected between the resilience principles and One Health approach. Even in a paper published in Uganda, and maybe Dr. Jonathan know about that, leadership is vital in the One Health approach for managing infectious disease, and particularly in the animal-human environment interface. Through modules like leadership in infectious disease management, participants from diverse disciplines in this situation, including public health and social sciences, were equipped with the skills to lead in collaborative cross-sectorial response. So the answer, how can the One Health approach enhance resilience? It could be in such way. The healthcare system resilience is the core, and then it's affected by human health. A human health can be affected by animal health, and animal health can be affected even by environmental health. So these are the categories, like the human health affecting health system and health professionals, and animal health. They are all reflecting and can affect each other. That's why we have an holistic approach to support One Health so that it can be helping resilience. Expanding the One Health approach could pave way for a more resilient and sustainable future for the healthcare system worldwide. Now, for the conclusion, adopting the One Health approach is critical in enhancing resilience, particularly regarding hospitals and healthcare systems. By integrating transformational leadership principles with the One Health approach, healthcare systems like One Health can build greater resilience against disasters and health crises. The integration empowers healthcare workers and fosters collaborative environment that addresses the complex interdependency between human, animal, and environmental health. Such a holistic approach is essential for creating sustainable health systems that adapt to future challenges. And finally, One Health strategy aims to address the root causes of disease emergence, not just the symptoms. Promoting environmental stewardship includes protecting forests, reducing pollution, and fostering biodiversity. Thank you so much. And these are the references and resources used. And thank you again.
Okay, so we'll have a couple or three questions according to, let me, okay, Rola Molam. The question is, how we can identify a newborn is diagnosed or portable of Mpox if no clinical signs seen on the newborn, and is placental swabbing can identify the Mpox infection in a newborn? I think Prof. Alfonso may answer this question. Prof. Alfonso is here, or?
Yes, I am here.
Yes, would you please answer this question? How you identify a newborn is diagnosed or carrying the Mpox if no clinical signs? We just want to know is the newborn is carrying or he is free or is free. So what's your opinion about this?
The first there will be echo if other mic is open. There is a lot of echo. I would say that it's important to consider that if you have a pregnant woman initially that is with clinical symptoms and is positive, regardless what is found clinically in the baby, you have to perform not only clinical screening but trying to test her, for example, in different liquids to try to detect if the virus is present. As I mentioned during my presentation, and other speakers also mentioned, we have to remember that we can detect the virus, not only the DNA of the virus, not only at the skin lesions, but also can be detected in blood, can be detected in other secretions, can be detected, as I mentioned, in sexual secretions and others in mucose. And it's important to remember that in that case, we have to screen because of the risk of transmission for the newborn. Additionally, while Fatma returns, additionally, it's important to remember that there have been reports of vertical transmission of Mpox, and even there are some interesting published systematic reviews regarding all the effects that has been documented recently, especially after the 2022 outbreaks regarding the impact of Mpox in pregnant women and newborn, and there's one that was published in one of the Lancet journals during that time.
Sultan Al-Nakhli, how about there's a nice question. Is there any reported reverse zoonosis cases? And if so, how the animal health authorities are dealing in contributing to the contingency task force? I think we have, there is something in, I think in 2023, there was a reverse zoonosis. A man transmitted monkeypox, a spillover to his pet. You remember this, Alfonso?
Yes, I would commend.
Yes.
In 2022, we start to discuss on that possibility regarding the possibility of transmission to domestic animals. We suspect that was possible considering different factors, and in fact, in August 2022, there was a report in Lancet regarding a HIV serodiscordant couple in France that transmit the same clade to their dog in Paris, and this was documented. But after that, Also in Brazil, in Minas Gerais, there was also another case. And after that, for example, in United Kingdom, they start to assess the risk of the reverse zoonotic transmission to pet animals in the midst of the epidemics in human. And in fact, for example, the CDC in United States also described the importance of the Mpox among pets in the home, and especially to avoid contact with animals if someone is infected with Mpox. And last year there started also an investigation in United States from the group of Morgan that was published in Emerging Infectious Disease that described precisely the detection of Mpox in dogs and cats and rabbits. And they found, for example, that from some there was especially positive in dogs.
So I want to ask something, Dr. Baddour, is there any reported, any recommendation for vaccination of a newborn to Mpox positive mother if no clinical symptoms is seen? Is there a recommendation to vaccine a newborn?
Up till now, the vaccines available are for adults. There are ongoing clinical trials for pediatric population vaccination. So I guess this will depend on the individual case-to-case scenario, so that there are no vaccines currently licensed for this age group. But maybe by mid-2025, the results of the clinical trials, some of them will be out, and maybe by then there will be vaccines suitable for such patients or such cases.
There is another question regarding the One Health approaches and strategies applied in affected regions. I don't know who wants to address that question.
What I mentioned is that the plan for response and preparedness that had been declared by CDC and WHO is always including several items that are in correlation with resilience and can be in correlation with One Health approach, So we can consider this is important because these actions that can be done, they are in correlation with both approaches.
There is another pending question from Kevin Ron. With resurgence of Mpox, how can we increase detection of a symptomatic case? What are the current challenges of identifying asymptomatic carriers, and how should future research of surveillance strategies to be designed to address potential signs of transmission change, especially among high-risk populations? I don't know if Professor Baddour would like to answer that question.
Okay. As mentioned during the presentation, even PCR would unlikely detect positive cases before the onset of symptoms. So we are talking about carriers or early onset cases where we don't really have symptoms yet. The disease has not yet shown on the persons. So it's not something easy to do. But what we can do is isolation of cases and surveillance with contact tracing. This has been very successful with smallpox and is recommended for Mpox as well. Case tracing. So whenever there's a case, all the close contacts of that case and the household contacts and even the healthcare workers that dealt with that case should be followed up and up to the incubation period. That is what we have now. I guess you might add something here, Professor Alfonso.
Yes, I only would like to add that in fact, although it's hard as Professor Baddour mentioned, it's important to remember that there exists asymptomatic infections. In fact, some studies have published about that, and in fact, during the epidemic in 2022, we published a systematic review about about the potential asymptomatic infection in vaccines, and we found that the pool prevalence of asymptomatic infections from 16 studies and just 645 individuals that were included there, it was 10%. So it's low, but may occur, and we have to keep that in mind, especially in high-risk populations.
Okay. Okay. Thank you for all of you for this very fruitful presentation, and thanks for all attendees. And I hope you got benefit from this webinar. Thank you, and looking forward to see you once more. Bye.
Bye. Thank you.
Bye. Thank you.